Hello, my name is Laura.
I'm Tommaso.
And we're in a group of Stanley and Nancy, and this is our presentation on DNA replication, transcription, and translation. And this is DNA replication. So the enzymes involved in DNA replication are DNA polymerase, primase, helicase, and ligase.
Okay, so first of all, the helicase, which goes across, as Laura will draw it, it cuts the DNA, it cuts the hydrogen bonds between the A, T, C, and G. After that, the DNA polymerase, the primase, has a primer, one of to the parent strand. So that the DNA polymerase knows where to start the DNA copy. After that, the DNA polymerase goes on both parent strands. Firstly on the 3' to 5', which goes in that direction, and then on the 5' to 3' going in that direction. It starts the copying. The complementary strand is created with the exact same DNA.
It can only go on a 5 to 3 direction. So because of this, on the 3 to 5 strand, they create open-market fragments because since it's only going this direction and it has to restart over, it leaves like this space between.
For that one, on the parent strand going 3' to 5', the primase also has to go back and set out a new primer every time for the DNA polymerase to restart the copying in that direction. So basically, for the DNA copying to move forward, on the 3 to 5' strand, the DNA polymerase has to go backwards to go forwards. If that makes sense.
And so another enzyme that is involved is the ligase, and it basically like glues the open fragments together so that there isn't like any issues with DNA.
And lastly, the topoisomerase is an enzyme which prevents supercoiling. So it basically goes ahead of the ligase before the DNA cutting occurs and releases tension between the two phosphorus strands that hold the DNA together.
Also, but the DNA polymerase also like checks over the entire process, so there's like barely any mistakes during DNA replication ever due to DNA.
Hi, I'm Stanley.
I'm Angela.
And this is
Transcription.
So, what's that next? Basically, the enzyme RNA polymerase binds to sites on the DNA at the start of a gene. A gene is just a sequence of DNA that's transcribed into RNA. So then RNA polymerase separates the DNA strand and synthesizes the complementary RNA copy from the antisense strand. So transcription happens in a 5 to 3 direction. So it adds the 5 end of the 3 RNA nucleotides to the 3 end of the growing RNA molecule. Obviously, it moves along in the 5 to 3 direction on the antisense strand. And it does this by covalently bonding the ribonucleoside triphosphates.
And the transcription happens in a nucleus. And after it transcribed into messenger RNA, you will pass through the support and go into the cytoplasm. And here's 2 types of gene expression. And this one is this one is prokaryotes.
This one is prokaryotes?
Yeah. So for this one, the RNA polymerase will attach to promoter, and once it attaches to this this open region, the transcription begins. And here's the repressor molecule. When the repressor molecule attaches to the operator, the transcription will stop. And for this eukaryotes,
so as in humans?
Yeah, human, for example. So in order for the transcription to start, the transcription factors have to attach to the RNA polymers and promoter. After these three attach together, the transcription will start.
Alright, so let's move on to translation now. So after the mRNA molecule is produced, it goes to the cytoplasm, but with going to the nucleopores. And then what the mRNA molecule does is it binds to the small subunits of the ribosome, and then the ribosome moves along the mRNA molecule in a 5 to 3 direction until it reaches a start codon AUG. So then the tRNA.
AUG is methionine.
Yes, exactly. So the tRNA carrying methionine, which is complementary to the start codon, exactly, binds to the P sites of the ribosome. Then what happens? Then the large subunit of the ribosome binds to the mRNA molecule and to the small subunit of the ribosome, and that's the whole part is called initiation. Then a second tRNA with its own amino acid, complementary to the second codon of the mRNA molecule, goes in and binds to the A site of the ribosome. And then what happens is that the amino acid carried in the P site is transferred to the amino acid of the tRNA on the A site, and the ribosome catalyzes a new peptide bond, which is a condensation reaction. And through this process, the polypeptide chain increases. Then the ribosome moves one codon along the mRNA molecule and in a 5 to 3 direction. And then what I meant is that the tRNA on the P site goes on the E site and then release, and the tRNA on the E site then goes to the P site. So then another tRNA binds, so it's not drawn here, but another tRNA binds because it goes like that, and it's complementary to the next one and the DNA, and it binds to the A site, so it goes through here. So you can see the pattern here, and then the last three steps I said are repeated continuously until a stop codon is reached. So then the amino acids bind to each other to the peptide bond, creating a polypeptide chain. So when a stop codon is reached, translation is stopped. So a release factor binds to the A site, the polypeptide chain is released, and the ribosome complex disassembles, ready to use for translating another RNA molecule.